right. Our next speaker for our recent graduate version of our series is Zach Lonergan. Zach recently graduated as well, and we're excited to hear from you, Zach, about your path, your personal journey, and what you're up to now. Thank you so much.
Sounds good. It's really good to see Rhonda, who was one of my IGP classmates. And we started Vanderbilt at the same time and we left around the same time. So yeah, it's good to see her. And thank you, Beth, for the invitation to talk today.
Yes, thank you.
The first thing I'm going to do is just briefly introduce where I am now. So I'm currently a postdoc at Caltech, and I defended my PhD in February 2020, so right before Rhonda. So this is a picture of me and my PhD advisor, Eric Scarr, at my defense party. And this was my going away party in the first week of March in Nashville, right before the world shut down, and we transitioned into a different kind of life for the last year. And so since then, after that picture was taken, I packed up my car and drove 3 days to California, and I lived in the state for 1 day before we went into statewide lockdown. So still getting to know the area, but this is the only photographic evidence of me in my current lab. So I'm currently a postdoc in the lab of Diane Newman, who's right here, and this was our lab photo for 2020. And I'll come back to this again a little later, but I want to sort of go back to the early days. So I'm originally from West Virginia, which is a pretty rural state in the Northeast. And I grew up in this mountain town named Elkins, which is right on the edge of the Monongahela National Forest. It's a pretty small town, it's very rural, and the whole state of West Virginia is the only state that's entirely encompassed within the region of Appalachia. So, we have rolling hills and lots of mountains, and it's very scenic. But I also grew up pretty close to this area known as the National Radio Quiet Zone. So it's a federally designated area in the mountains of West Virginia where there's limited radio activity, including things like cell phone service or even like radios for your car. And they have little trucks that drive around and try to ping for rogue radio waves, because the government runs these really large steerable radio telescopes in the mountains that capture trace radio waves from outer space. So they're so sensitive that any like rogue radio waves can be picked up by these satellites. So there's this entire zone known as the National Radio Quiet Zone that's pretty close to where I live. So because of that, it actually acts as a great preservation system, right? So it's pretty pristine. Lots of hiking and outdoors time, which is, I would say, how I spent a majority of my childhood. It's a little bit of a negative, because it is very rural, and there are, because of that, all the negativities that come with living in a remote part of the country. 3 hours to the closest airport, even though it's right in the middle of all these other northeastern states. But I really credit like the opportunity for me to be outside and to just take in nature as being something that sort of drove me to ask, how does the world work? And I think what led me to science eventually. So I was like definitely a high school science nerd. I didn't really know a lot about science beforehand. I come from a really big family, so I'm one of 7. So there wasn't like a lot of time at home aside from like helping out with my siblings or helping run errands to do other extracurriculars. But I really loved science. I remember the first time I like colored in this diagram of the cell was like probably one of the most eye-opening experiences of my life. And you learn about DNA and base pairing, and I was like, wow, this is the coolest thing ever. And I, at that point, the only visible career path, and I think this is similar to what Rhonda said, is like, if you're a smart kid in high school and you're good at science, like you should just go to medical school. That's really the only visible career I saw as a path forward in science. But I didn't really know what I wanted to do at this point, obviously, by any stretch of the imagination. I was involved in some other stuff in high school. So I did athletics and I was in student government. But I also, I worked part-time. So I worked at McDonald's throughout my high school tenure. It was like a requirement by my parents that if I wanted a cell phone, I had to be able to get a job and buy the phone with my own money. So it was definitely one of the most rigorous jobs I think I've ever had, and I was there for like multiple years, and it was actually a really good time, and I was there with all my friends from high school. But um, yeah, this was my high school experience in a nutshell. DNA and McDonald's, right? Um, so I had this vision that maybe I wanted to go to medical school, and so I looked at colleges in the area that had pretty good placement rates for that. And there was a local liberal arts college called West Virginia Wesleyan College where I did my bachelor's degree. And I was a biology major, and I came in and I was, I took all the science classes that I loved. But then I heard about this research opportunity with a new faculty member at Wesleyan who worked on bacteria, and I approached her after my freshman year and I was like, hey, I would really like to work in your lab. I don't really know what that encompasses, but it seems like something that could be fun. So she let me work in her lab, which was my first research experience. And luck would have it that around that time, the college was updating their publicity for the website so I got a little candid shot of me in the lab pipetting that is still on the college's website. So it's a nice trip down memory lane when I'm clicking through and I see my face pop up. But my first experience was with Dr. Melanie Saul, who is still a biology professor at the college, and she worked with this bacterium known as Borrelia burgdorferi, which is the bacterium that causes Lyme disease. So it's transmitted through ticks, and they're really strange-looking bacteria that form these long wave-like cells. And when you get bit by a tick that's infected with Borrelia and it transmits it to you, you sometimes develop this really characteristic bullseye rash. So we were studying the morphology of Borrelia, and it was a really great experience for me to just sort of get in the lab and learn the basics at my small school. The research opportunities, though, at a small liberal arts college are very different than if you're at a big university. So I was encouraged to seek out other research experiences similar to VSSA or things that happen at other places. So during my undergrad years, I did a number of different experiences. The first one was through a program sponsored by the state of West Virginia called the West Virginia INBRE program. And I worked with Dr. Laura Gibson at West Virginia University. And this was a little bit different than my other projects. So it was a cancer immunology lab. And this is really my first foray into like what it means to be a scientist and to do research. And it was like super hard. I had never had any immunology classes or microbiology classes at that point. So like the learning curve for the first few weeks was really intense, but overall it ended up being a really awesome experience. And I ended up being like super prepared for my next academic year because I learned like what a B cell and a T cell was the summer before. But I continued my research at Wesleyan during the academic year, and then the following summer, I did another research experience through the NSF, through the research experiences for undergraduate programs, and it ended up being back at WVU, where I worked with Jen Gallagher, and she's a professor there that studies. She uses yeast as a model organism to study physiology and their responses to intoxication. Similar to Rhonda, I actually did have the opportunity to study abroad as well. So this was something that I think I had been told wasn't very possible if you're in a STEM major, but I think, if it's something that you think is important, there's definitely ways to make it happen. And it ended up being one of the most rewarding experiences in my undergrad career. So I spent a semester at Queen's University in Belfast, and I think what was a little unique about my experience is I still had like a lot of science classes, obviously, that I needed to fulfill for my degree, but I was able to enroll in courses there that fulfilled the requirements that I had back home. So part of that was taking a field ecology course where I spent a few weeks in rural Ireland, and we did this technique called flagging, which is what this person's doing here, where you take basically a white sheet, and you can use this as a technique to collect insects, but we were specifically doing it to collect ticks. To look for the presence of Lyme disease in Northern Ireland. So it was sort of a field experience parallel to the lab experience that I had at my undergraduate institution, which was really fun. Aside from that, I was able to take classes that I never would have been able to take in my college. So I took a marine biology course, and it included trips to the coast and a parasitology course. So it really rounded out my love of all things microbe and all things, disease. So then, of course, the existential question that I had to face when I was in undergrad was like, do I go to med school? Do I go to grad school? And I think it became pretty obvious to me that I really enjoyed the the discovery associated with science, and I liked learning about the disease perspective in a way that I thought was unique to graduate school that I wouldn't necessarily get from medical school. So I didn't really know what kind of research I wanted to do because I had had a few different research experiences. So I only looked at umbrella type programs where, you know, I could rotate in many different kinds of departments for um, uh, for college. So I ended up at Vanderbilt in the interdisciplinary graduate program. And I have to say it was really great experience. So I joined the lab of Eric Scarr, who's right here. And this was our lab photo in 2019. And yeah, overall, I had a really positive experience at Vanderbilt. I had interviewed at other places, but I chose Vanderbilt for a number of reasons. One was because Nashville was a growing city and a really exciting city, and the students genuinely seemed happy. And I was looking at universities in other cities that the stipend was like the same as what it was as Vanderbilt, but it was 3 times the cost of living. So I was able to actually live in Nashville and enjoy myself and strike a really good work-life balance, which was something I was looking for in a graduate program. My PhD focused on this opportunistic bacterial pathogen named Acinetobacter baumannii. So you can see it here at these rods in contact with some lung cells. And most of my PhD work was sort of understanding how these bacteria are able to survive inside human hosts, and we used animal models to assess that. And during my time working on Acinetobacter, the World Health Organization came out with a list of pathogens that they deemed to be really critical for more research. And for multiple years in a row now, Acinetobacter has been the #1 critical pathogen as defined by the WHO, for which we need new therapeutics. And the main cause being because a lot of strains of Acinetobacter are resistant to antibiotics. So there's an underlying need for more drugs to treat these infections. So it was rewarding, at least from a research perspective, to know that I was working on something that maybe had some relevance to real world problems, which I liked a lot. So similar to Rhonda, it was more than just being in the lab when I was at Vanderbilt. So my lab mates became great friends, and we did a lot of really fun things together. We had holiday parties together. We went on races together, and these are people that I still talk to on a regular basis. That enriches the graduate school experience in a way that I think is really important for you to make the experience what you want it to be. And here's my dog Maya right here. She's really sweet. So I finished up my PhD, and I didn't know when I started graduate school that I wanted to do a postdoc or I wanted to stay in academia. I knew I really liked science, and I knew that I liked research, and it happened to be that during my time in grad school, I had a really positive experience meeting other faculty members and seeing the kind of work they do and the kind of groups that they were able to lead. And I was like, this might actually be something that I'd really like to do. So I think it was probably in my 4th year of graduate school that I decided I wanted to do a postdoc in a type of position where I could hopefully eventually run my own lab. So I interviewed at a few different places, and I eventually ended up in the lab of Diane Newman, who's a professor in biology and in geobiology at the California Institute of Technology in Pasadena. So Pasadena is, I don't know if I even knew where it was before I interviewed, but it's about 10 miles northeast of Los Angeles. So it's sort of a suburban city right on the edge of this sprawling metropolis. And Diane's and the lab's research really broadly focuses on how bacteria are able to survive in really diverse environments. So part of the lab studies how bacteria survive in soil environments, and another arm of the lab that I'm involved with studies how bacteria are able to survive in chronic infections and in low oxygen environments. So the question that I sort of wanted to pursue during my postdoc is something that I've been thinking about for a while, since my undergrad years working with Lyme disease pathogens, is that what makes some bacteria deadly? So the Nobel laureate Francois Jacob famously said that the dream of every cell is to become 2 cells, right? So this is true for bacteria, they want to multiply, but this is also true for high organisms. This is true for vertebrates. This is true for humans. And when you consider this paradigm, you can think about what is the difference between a single cell or a human to become more than one. And if you consider one of these to be a pathogen, what does it mean for the pathogen to become more than one if it has to happen within the human? This is a really interesting conundrum, right? And I think we know a lot about certain pathogens and how they were able to replicate and cause disease, but there's a lot we don't understand in certain models where it's not really obvious. One case where it's really not obvious is in the case of people that suffer from cystic fibrosis. So opportunistic pathogens in cystic fibrosis is a really big problem. Cystic fibrosis itself is a genetic disorder that affects about 30,000 people in the US alone. And it's characterized by whole body disease symptoms, but most notably disease symptoms in the lungs. So on the left here is a picture of airways in a healthy lung, and you can see they're very clear and allow the passage of air in and out for gas diffusion and breathing. What happens in the case of cystic fibrosis, though, is there's a genetic defect that leads to accumulation of these really thick mucus layers within the linings of the lungs. It restricts airway movement and restricts breathing, but it also serves as a site where bacteria can colonize people and form these bacterial infections that are chronic and persist for the entire life of the person. And unfortunately, a majority of people with CF actually succumb to these bacterial infections. And while the life expectancy has been increasing for people with CF, it's currently about 40 years, even in developed countries. So sort of at a molecular level, what we know happens is that in healthy airways, like I just showed you, if we zoom in, there are these ion channels that aid in maintaining osmotic homeostasis at the air cell interface. But in the case of cystic fibrosis, there's a mutation in one of these transporters known as the CFTR that basically prevents proper osmotic homeostasis from being maintained. It leads to this mucus production. And within this mucus, in pink and purple here, we have these immune cells that come in because of the inflammation, but they're unable to clear these infections, which are described here. So I was really interested in like, how do these infections form and what are there ways that we can understand them at a molecular level? Something that's unique about cystic fibrosis too is that not every bacterial species can infect a person with cystic fibrosis. So people have surveyed the causative agents of these lung diseases, and there's really a small clustering of all microbes that cause these diseases. So what I'm showing here is the relative percent of people with CF that have certain bacterial species in their lungs, and then their age on the x-axis. And basically what I want to show you here is that the species necessarily don't really matter, but there are trends that happen as someone ages, where one species, for instance, in this green line is elevated when people are young, but decreases over age, while this one in the blue line is low early in age, but then increases later in age. What we don't understand are the dynamics that regulate these changes in the microbial community and how they relate to the disease progression. So this was really a question that I wanted to address in my postdoc, is what drives these changes in the composition over time in someone with cystic fibrosis? And if we understand this, is there a way that we can target it with new drugs? So the plan was, when I started my postdoc and we spent some time developing this, is that we were going to collaborate with the University of Southern California, which has a clinic to treat people with cystic fibrosis. And we can collect basically the fluids and microbes from the lungs of these patients when they come in for routine care. So we would collect these samples, and then through a number of processes, we would purify them and then analyze them in a number of different assays. So I'm not going to go into what these are, but we have ways that we can figure out the community structure as well as some of the physiology and properties of these microbes. The reality, though, set in pretty obvious that in the first few months into 2020 that this wasn't going to happen the way we wanted to. And this was, of course, mainly driven by the COVID-19 pandemic, which limited the ability for us to have this workflow for a number of reasons. First of all, is that the CF clinic was working on a limited capacity because they wanted to keep density low. And also people with cystic fibrosis are immunocompromised, so they are already a high-risk population for susceptibility to COVID-19. So there weren't a lot of patients coming into the clinic, and then we found out later that the patients that weren't coming in and getting care typically were because they also had COVID-19 infections in their lungs as well. So we didn't really want to bring COVID-19 and our bacterial samples into the lab, so it pretty much prevented us from completing this workflow at all. And it was a really frustrating experience because I had spent a lot of time figuring out the way that we could get these samples and the ways that we could process them and analyze them to ask some of these questions I was interested in. But what we did instead was I worked with my mentor, and we sort of went back to a more reductionist approach. And I go back to this quote of the dream of every cell is to become two cells, because what we ended up doing is focusing on a few of these species that we know cause cystic fibrosis disease, understand the mechanism of how they are able to grow in really hostile environments, and hopefully move that into a system where we can understand how they actually infect these lungs. So it's sort of the precursor steps to the actual disease, and working from the front instead of from the back, like we were planning to do for my original project. And this is sort of still in its infancy. It's only in the last few months that we've even gotten back full time in the lab because of the pandemic. But I think it was a really good lesson into being flexible with science and with the path forward that we take. And so I think overall, my time so far as a postdoc and through graduate school and before has been, if you can't embrace uncertainty, is to at least accept that, the plans are going to change, and that the route you take is not going to be linear, right? So we're on our own path, and what's right for you is not going to be right for someone else. What's really important to me that I learned is that one step will inform the next. And so you don't have to have a plan for the next 5 years or even 1 year, right? You only need to have a plan for the next, sometimes a week. And then you take that step and you see where things are and you reevaluate, and then you take the next step. And eventually, before you know it, you've taken 100 steps and you're in a new place. And the mistakes and failures are like totally inevitable. And this is going to be true for any career. So it's definitely true in research. It's definitely true in academic research. A lot of things you try aren't going to work out, but it's important to have a plan B through Z. And you take one step and you inform the next. And part of that is adapting to the failures that are true in life. Something else that I also learned, probably through the hard way, because I like to just do things on my own and try to figure them out, is you always want to ask for help. So it's important early on to build and rely on a support network. This can be your advisor, it can be your friends, or it could even be other people who are working in the field related to you. So sometimes if you don't know the answer to the question and you want to, Google is really an amazing resource, and you can learn. I mean, I think like 75% of what I've learned has just been from like a Google search and just retaining some of that information. So don't underestimate the power of your own ability to use the internet. And I think also you want to seek out advice whenever you can. You want to ask people that know your work, that know how you think, and that can help make you a more informed person. And the last thing that I think is really important, and I definitely try to do, is you want to find new outlets for your science too, and you want to have a holistic experience in whatever career you have. So for me, I found that I really like mentorship as well. So I'm mentoring an undergrad student this summer in the lab, and it's been like so rewarding just teaching him how to learn about the basics of science, learn how to work with bacteria, and it teaches you to appreciate all you've learned to get to where you are at each step. I also think that finding a way to get back and using your knowledge of science through community involvement and volunteerism is a really rewarding way to use your scientific knowledge to sort of better the world in your own small way. And finding new hobbies is also a great outlet. So something like baking, like Rhonda mentioned, or I took up running in graduate school, and it was like a great way to be social with my lab mates and also sort of channel some of my anxiety when things weren't working out in the lab. So finding new hobbies and finding ways to keep yourself healthy are great ways to sort of manage a life in science or in any career. And with that, I'm happy to take any questions. I'm on Twitter, and you can also find me on LinkedIn as well. So thanks.
Wow, thank you so much, Zach. That was really, really awesome to hear your journey and all of your steps. I'm sure we'll have a couple of questions pop up. I've got one real quick. So you said you may not always need to know what your 5-year plan is, and one step at a time, knowing what that next step is the important thing. When you started grad school, or maybe even when you were making this debate about med school versus grad school, did you know that you would want to be a faculty member? Or, I mean, you seem to have a pretty good sense of that now. Did you have even any specific career plans in mind at that point?
Yeah, so I was actually really interested in science policy when I was interviewing for grad school. I didn't really know what that meant, but I was interested in sort of how government influences what we do in the lab. But then once I sort of got a sense for the work that actually is involved in that kind of career, I decided it wasn't really for me. And for a while, I didn't really know what I wanted to do. So, I went to some of the Brett events at Vanderbilt to learn about career options. But it was really, for me, it was meeting other scientists and meeting um, mentors and advisors who are in positions where they really love their job. Because I think a lot of times you hear about all the negatives of academia, and it's so hard to get a job, and it's you have to write grants all the time, and you have to like you're always like one step away from your lab closing. And I think that is true sometimes, but there's also a lot of pros that come with the career. And it's I was advised, don't let the statistics make you fearful of a career if you think it's a good fit. So once I sort of learned that, I began to embrace the idea a little bit more. But yeah, it was definitely not something that I was really on my radar when I started grad school, and it was a progression over many years.
In my experience, seeing me and my classmates, I think that the statistics of how many people get a faculty position, it's because other people just find what they love.
Mhm.
I do think it's
Yeah.
competitive. That's not untrue. But I also find that just so many of the students, I don't want to say leave that path, because it's not that, it's that they find their own path rather than feeling like they have to stick to this one thing that's the only thing you should do. That's just a misrepresentation of reality.
Yeah, absolutely. There are many great careers and many ways to be happy in life. So this is one option that I found work for me.
Great. Any other questions? All right. Well, thank you so much for being here. Thank you, Zach. Thank you, Rhonda. Thank you, all the audience. We'll see you next time. Next week, we'll do individuals who are in their career on their like fairly stable path. So we'll get going there. Thank you again. Good to see everybody.
Check out our website to find recordings of all of our videos. We'll get them posted about a week after each of the presentations. We can't wait to see you!